Good morning, this is Jill Wilson with Cedar Mountain Perennials, and today we're going to talk about seed germination strategies. And one of the very interesting and actually one of the tricky things about growing native plants is that they employ a variety of strategies that call for different triggers for seed generation. And there's a wide variety of reasons for this. One of them is that if seeds mature in the fall and the plant has to undergo a difficult winter, cold winter, then the plant needs to evolve methods so that those seeds don't germinate in fall with the first moisture that they carry through the winter before germinating. Another strategy involves what's called seed banking, where plant produces seed, they go into the ground, and in order to prevent all the seeds from germinating at once following favorable conditions, they will have things like a very hard seed coat, which takes a while to break down. And what this does is it means that not all the seeds will germinate at the same time. And this is beneficial for the plant because it could be that you have a drought year or you have other conditions that are not favorable for growing, and if all of your seeds don't germinate at one time, that's going to be better off overall for the plant. So let's look at some examples here. We employ different methods of stratification, primarily different lengths of cold, also different types of external seed coat treatment, such as nicking the seeds with a knife or razor blade actually, or exposing them to hot boiling water or an acid treatment. We also may actually plant the seeds and then put them outside, in which case, as opposed to refrigerator, which is our normal treatment. And the reason for that is that when they are outside, they get exposed to a warm-cold regime, as opposed to just in a refrigerator where it's a constant cool temperature. So let's look at the effects of this. This seed tray is of what we call sticky geranium, Geranium viscosissimum. It's a local native, and as you can see, there's almost no germination in this seed tray. We plant 100 seeds in each of our seed trays, and then we keep track of how many germinate so that we can evaluate the effectiveness of our seed treatments. So as you can see, this one has very little, and this particular tray, seeds were planted, but they were planted and there was no subsequent seed treatment. So no cold, no nicking of the seeds, no boiling water, nothing. Now, in this seed tray, you can see that we have quite a bit of germination. This is also sticky geranium. And if I look at this tag, in which we've described this, we have the species, where the seed lot number, which is where they were collected, and then the date that they were treated. And so these seeds, which were treated in March and then placed out in warm temps, these were scarified. Doesn't say hand scarification, although I'm guessing that's the case. So probably we took a razor blade to these seeds to nick the seed coat, which then allows the seed coat to take in water. Now, here's another tray, also of sticky geranium, and you notice that this one has good germination, and let's see what the treatments were here. This says that these seeds were exposed to 1 month of cold in a refrigerator. So both of those treatments look to be pretty effective for germinating seeds of geranium. Okay, now this is northern vetch or boreal vetch, Hedysarum boreale, and you can see this tray, which has some germination, but not spectacular germination. This is 100 seeds. And these seeds were exposed to hot water or boiling water as a seed treatment. Okay, let's look at this tray. This is the same species, so this is also boiled vetch, and you can see excellent germination in this tray. And these seeds were hand scarified. So these seeds were nicked with a razor blade prior to being planted and placed out here with favorable temperatures. Okay, this is yellow columbine. It's our local native columbine, and it grows up in the mountains here in North Idaho. And as you can see, I can just see 2 seedlings in this tray, and these seeds were scarified, so a hand treatment. This tray is also yellow columbine, Equilegia flavescens, and as you can see, it has much better germination. And these seeds were put in the refrigerator for 3 months, so 3 months in the refrigerator. Last example that I'll share, this is Phlox diffusa, or creeping phlox. It's another native species here in North Idaho, and you can see just a few seedlings. And these were exposed to 3 months cold in the refrigerator. Same species, Phlox diffusa, and you can see many more seedlings. And these were sown out or placed outdoors over the winter. So these seedlings would have been exposed to variable temperatures, so as opposed to the constant temperature that might be found in a refrigerator. So as you can see, growing native plants is not as simple as just throwing some seeds in a tray and waiting for them to germinate. And it's particularly so when there may not be information about a particular species about the ideal conditions for germination. So that means that those of us who grow native plants must keep careful records and try a number of different techniques to learn how best to grow their plants. So lots of science in addition to the art of growing native plants. Thanks for joining me. I'm Jill Wilson with Cedar Mountain Perennials. Thank you.